Apoptosis is a form of programmed cell death that occurs in multicellular organisms and in some eukaryotic, single-celled microorganisms such as yeast. Biochemical events led to characteristic cell changes, morphology, and death. These changes include blebbing, cell shrinkage, nuclear fragmentation, chromatin condensation, DNA fragmentation, and mRNA decay. Apoptosis in etoposide-treated Du-145 prostate cancer cell exploding into a cascade of apoptotic bodies. The sub-images were extracted from a 61-hour time-lapse microscopy video, created using quantitative phase contrast microscopy. The optical thickness is color-coded. With increasing thickness, color changes from gray to yellow, red, purple and finally black. In contrast to necrosis, which is a form of traumatic cell death that results from acute cellular injury. Apoptosis is a highly regulated and controlled process that confers advantages during an organism's life cycle. For example, the separation of fingers and toes in a developing human embryo occurs because cells between the digits undergo a form of apoptosis that is genetically determined, unlike necrosis. Apoptosis produces cell fragments called apoptotic bodies that phagocytes are able to engulf and remove before the contents of the cell can spill out onto surrounding cells and cause damage to them. Because apoptosis cannot stop once it has begun, it is a highly regulated process. Apoptosis can be initiated through one of two pathways. In the intrinsic pathway, the cell kills itself because it senses cell stress, while in the extrinsic pathway the cell kills itself because of signals from other cells. Weak external signals may also activate the intrinsic pathway of apoptosis. Both pathways induce cell death by activating caspases, which are proteases, or enzymes that degrade proteins. The two pathways both activate initiator caspases, which then activate executioner caspases, which then kill the cell by degrading proteins indiscriminately. In addition to its importance as a biological phenomenon, defective apoptotic processes have been implicated in a wide variety of diseases. Excessive apoptosis causes atrophy, whereas an insufficient amount results in uncontrolled cell proliferation, such as cancer. Some factors like FUS receptors and caspases promote apoptosis, while some members of the BCL2 family of proteins inhibit apoptosis. Activation mechanisms. Control of the apoptotic mechanisms. The initiation of apoptosis is tightly regulated by activation mechanisms, because once apoptosis has begun, it inevitably leads to the death of the cell. The two best understood activation mechanisms are the intrinsic pathway, also called the mitochondrial pathway, and the extrinsic pathway. The intrinsic pathway is activated by intracellular signals generated when cells are stressed and depends on the release of proteins from the intermembrane space of mitochondria. The extrinsic pathway is activated by extracellular ligands binding to cell surface death receptors, which leads to the formation of the death-inducing signaling complex, DISC. A cell initiates intracellular apoptotic signaling in response to a stress, which may bring about cell death. The binding of nuclear receptors by glucocorticoids, heat, radiation, nutrient deprivation, viral infection, hypoxia, increased intracellular concentration of free fatty acids and increased intracellular calcium concentration, for example, by damage to the membrane, can all trigger the release of intracellular apoptotic signals by a damaged cell. A number of cellular components, such as poly-ADP ribose polymerase, may also help regulate apoptosis. Single-cell fluctuations have been observed in experimental studies of stress-induced apoptosis, before the actual process of cell death is precipitated by enzymes. Apoptotic signals must cause regulatory proteins to initiate the apoptosis pathway. This step allows those signals to cause cell death, or the process to be stopped. Should the cell no longer need to die, several proteins are involved, but two main methods of regulation have been identified: the targeting of mitochondria functionality, or directly transducing the signal via adapter proteins to the apoptotic mechanisms. An extrinsic pathway for initiation identified in several toxin studies is an increase in calcium concentration within a cell caused by drug activity, which also can cause apoptosis via a calcium-binding protease calpam. Intrinsic pathway. The intrinsic pathway is also known as the mitochondrial pathway. Mitochondria are essential to multicellular life. Without them, a cell ceases to respire aerobically and quickly dies. This fact forms the basis for some apoptotic pathways. Apoptotic proteins that target mitochondria affect them in different ways. They may cause mitochondrial swelling through the formation of membrane pores. Or they may increase the permeability of the mitochondrial membrane and cause apoptotic effectors to leak out. There is also a growing body of evidence indicating that nitric oxide is able to induce apoptosis by helping to dissipate the membrane potential of mitochondria and therefore make it more permeable. Nitric oxide has been implicated in initiating and inhibiting apoptosis through its possible action as a signal molecule of subsequent pathways that activate apoptosis. During apoptosis, cytochrome C is released from mitochondria through the actions of the proteins Bax and Bak. The mechanism of this release is enigmatic, but appears to stem from a multitude of Bax, Bak homo and heterodimers of Bax, Bak inserted into the outer membrane. Once cytochrome C is released, it binds with apoptotic protease activating factor 1, APAF1, and ATP, which then bind to pro-caspase 9 to create a protein complex known as an AP optosome. The AP optosome cleaves the pro-caspases to its active form of caspase 9, which in turn cleaves and activates pro-caspases into the effector caspase 3. Mitochondria also release proteins known as SMACs, second mitochondria-derived activator of caspases, into the cell's cytosol following the increase in permeability of the mitochondria membranes. SMAC binds to proteins that inhibit apoptosis, IAPs, thereby deactivating them, and preventing the IAPs from arresting the process and therefore allowing apoptosis to proceed. IAP also normally suppresses the activity of a group of cysteine proteases called caspases, which carry out the degradation of the cell. Therefore, the actual degradation enzymes can be seen to be indirectly regulated by mitochondrial permeability. Citation needed. Extrinsic pathway overview of TNF, left, and FAS, right, signaling in apoptosis. An example of direct signal transduction. Two theories of the direct initiation of apoptotic mechanisms in mammals have been suggested. The TNF-induced, tumor necrosis factor, model and the FAS-FAS ligand-mediated model, both involving receptors of the TNF receptor, TNFR, family, 39, coupled to extrinsic signals. TNF pathway TNF-alpha is a cytokine produced mainly by activated macrophages, and is the major extrinsic mediator of apoptosis. Most cells in the human body have two receptors for TNF-alpha, TNFR1 and TNFR2. The binding of TNF-alpha to TNFR1 has been shown to initiate the pathway that leads to caspase activation via the intermediate membrane proteins TNF receptor-associated death domain, TRADD, and FAS-associated death domain protein, FADD. CIAP1/2 can inhibit TNF-alpha signaling by binding to TRAF2. FLIP inhibits the activation of caspase 8. Binding of this receptor can also indirectly lead to the activation of transcription factors involved in cell survival and inflammatory responses. However, signaling through TNFR1 might also induce apoptosis in a caspase-independent manner. Better source needed. The link between TNF-alpha and apoptosis shows why an abnormal production of TNF-alpha plays a fundamental role in several human diseases, especially in autoimmune diseases. The TNF-alpha receptor superfamily also includes death receptors, DRs, such as DR4 and DR5. These receptors bind to the protein trail and mediate apoptosis. Apoptosis is known to be one of the primary mechanisms of targeted cancer therapy. Luminescent iridium complex peptide hybrids, IPHs, have recently been designed, which mimic trail and bind to death receptors on cancer cells, thereby inducing their apoptosis. FAS. The FAS receptor, first apoptosis signal, also known as APO1 or CD95, is a transmembrane protein of the TNF family which binds the FAS ligand, FAS-L. The interaction between FAS and FAS-L results in the formation of the death-inducing signaling complex, DISC, which contains the FADD, caspase 8 and caspase 10. In some types of cells, type 1, processed caspase 8 directly activates other members of the caspase family and triggers the execution of apoptosis of the cell. In other types of cells, type 2, the fast disk starts a feedback loop that spirals into increasing release of propoptotic factors from mitochondria in the amplified activation of caspase 8. Common components following TNFR1 and FUS activation in mammalian cells, citation needed, a balance between propoptotic, BAX, 46, BID, BAC, or BAD, and anti-apoptotic, BCLXL and BCL2, members of the BCL2 family are established. This balance is the proportion of propoptotic homodimers that form in the outer membrane of the mitochondrion. The propoptotic homodimers are required to make the mitochondrial membrane permeable for the release of caspase activators such as cytochrome C and SMAC. Control of propoptotic proteins under normal cell conditions of non-apoptotic cells is incompletely understood, but in general, BACs or BAC are activated by the activation of BH3-only proteins. Part of the BCL2 family.